electrical transduction channels. Now, the structure of the outer hair cell is also very, very interesting. They're just a little bit different than other types of cells. Generally, every cell has a cytoskeleton that is there to maintain the shape of the cell. But for the outer hair cells, that cytoskeleton is more small and it is more toward the top of the hair cell. And there is a reason for that. There is a pretty good reason for that, as well as we're going to talk about here in a little bit. Now, the cytoplasmic membrane of the outer hair cells has also its own characteristics. It has a lot of or high concentration of proteins, which makes it pretty rigid and stiff, which is important because, as we're going to see, those outer hair cells, they do their function through changing their length. And it is important, if they're going to contract and elongate, if they're going to change their length, it is important that their structure is going to allow them to do that. So they shouldn't be bending, or they shouldn't have the cytoskeleton that will prevent them from changing their length. Now, one of the proteins that is present within the cytoplasmic membrane is the protein pristin, which was described back in 2000 by Dallas and his group in this study with Zhang et al. And it was identified as the protein that is potentially responsible for the outer hair cells changing their length, or having the function of electromotility, or that movement. And protein pristin is one of those anion transport motor proteins, which is does its job basically through sensing changes within the hair cell, and uses that through movement of the anion chloride. Still continuing talking about the structure of the outer hair cells. The outer hair cells are supported by one of those supporting cells called the Dater cells. Those Dater cells over here, or in this image over here, it forms like a cup holder. So basically, the outer hair cell is not supposed to be in contact with anything at the bottom. There is no cell to hold it in place, because the idea here is the outer hair cell, to be able to do its job, it needs to contract and elongate, and it shouldn't be tied down by anything. Now, the other thing about the Dater cells is they have those phalangeal processes which are extending all the way to the top to help with forming that reticular lamina, which is the important structure that's keeping the charge for the fluid inside the cochlea. The space of null is where that gaps between those hair cells where the Corti lymph is basically filling. Now, the organ of Corti has also another important cell structure, the inner hair cells. The inner hair cells are arranged in one row, down, back over here. And on average, we have close to about 3,500 cells within that row. They're a little bit different than the outer hair cells. They're more flask-shaped cells, and on average, they are about 35 micrometer in length. Their stereocilia is also arranged in more of a U-shaped organization. And they have also changes in the number of stereocilia available for each cell. So at the apex, those hair cells, each hair cell has about 20 stereocilia, but as we go more toward the base, the number of stereocilia increases to a little over 50. Now, one important thing over here, and I kept it over here, the Henson strip. Now, the outer hair cells, their stereocilia is embedded within the tinctorial membrane. But for the inner hair cells, their stereocilia is not in contact with the tinctorial membrane, but there is that little groove that runs on top of where the hair cells are located. Now, talking about the inner hair cells, and as we're going to see in a little bit, the inner hair cells are an important structure, and part of their role is to send the signal down to the auditory nerve. So those inner hair cells, they have interesting synapse. It is called the ribbon synapse, which is this one here. This is called the ribbon, which is a pretty large organelle. And this organelle, it basically has the vesicles that are filled with glutamate, which is the excitatory neurotransmitter that is released by the inner hair cells. They have tethered vesicles, and they're going to guide those vesicles down to be released when there is a stimulation coming through. Now, as we can see also around this organelle, there is a bunch of other vesicles that are floating, and they're waiting for their turn to be tethered to the ribbon synapse. Each ribbon holds up to about 100 vesicles at a time, which we can imagine the number of neurotransmitters that they're able or capable of releasing. Now, that's generally the anatomical overview of the organ of Corti. Now, the question is, how does it all work? How does it all come together to generate a signal that's going to go all the way through the auditory nerve to the brain? Now, the cochlear structure, or the cochlea, has important characteristics that's going to allow it to do its job. So looking at the cochlea here, if we go ahead and unroll the cochlea, there is the scala vestibuli, there is the scala tympani, and this is our cochlear duct. This is our scala media. Now, over here, we're looking, this is the oval window where the stapes is going to sit there. So when the sound is coming through the ear canal, coming through the ossicles, it's going to get to the stapes, and the stapes is going to start pushing and pulling on the oval window membrane to send the acoustic signal inside. Now, as we mentioned, the cochlea, or the inner ear in general, basically, it's a fluid-filled structure. And fluid is not compressible. So if we're trying to apply force, if we have the vibration, the stapes is pushing on that fluid, it needs to have a little bit of a path to. Is there a question? So as the pressure pushing down, it needs to find a little bit of a pathway to get out. So that's where we have the round window with the round window membrane. When we push in with the pressure, when we push in on the oval window, that pressure is going to go through, and it's going to push the fluid, and find a path to go out here. That's basically a big factor why, for example, the sound is going to go all the way down here because we have a little bit of a path that's going to allow the fluid to move. With that being said, something to keep in mind, if we have a situation where we have a third window inside the inner ear, like for example with the superior semicircular canal dehiscence, and now we have another potentially easier path for the fluid to move to because of the presence of a low membrane, low window by the superior semicircular canal, that energy is not going to go all of it. It's not going to go all the way down. It's going to leak to the vestibular system, and we're going to end up seeing a little bit of a hearing loss. Now, the thing is, what we're trying to say is, there we go. When the vibration is happening, there we have the stapes pushing on the oval window, and that is pushing the fluid, and it's coming back to the round window. Now, all of this was described back in 1960 by von Beckersey when he described the traveling wave and described how the energy as it travels, it is going to peak at a specific point on the basilar membrane. And he was able to observe what's happening in cadavers, looking at different frequencies and where they're going to peak on the basilar membrane. So at 250 hertz, we can see this is more toward the apex. 1000 hertz, this is more toward the middle, and then the higher frequencies are going to be closer to the base. I wanted to put this one here as well. This is a click stimulus and how the click stimulus forms its traveling waves across the basilar membrane, because click stimulus is a broad stimulus that's going to run through, and it is going to basically activate throughout the cochlea. So what is happening? When we have an acoustic stimulus coming through, there we have the tuning fork running, creating those rarefactions and condensations, the increase in pressure and decrease in pressures. Those condensations and rarefactions are going to hit that tympanic membrane and start pushing either in or pulling it out, because when we apply pressure with a condensation, if higher pressure on this side of the tympanic membrane, it is going to push it inward. If there is a rarefaction hitting over here, that's a lower pressure, so it's going to pull the tympanic membrane outward. And that is happening back and forth, sending the signal. Now, these compressions and rarefactions, they're taking all the way to the oval window. So this is the membrane of the oval window as it is basically stimulated by the stapes, the foot plate of the stapes. When the sound is coming in, when there is a high pressure, so it's going to push it in. When there is a lower pressure, it's going to push it out or pull it out. So as a result, we're going to see this is happening. So looking at this one, I really like this demonstration because it is showing us that the whole cochlear duct is moving up and down, because what's happening when the pressure is coming through, it's affecting the mechanics of the whole structure. It is increasing pressure over here or over here, and as a result, it's going to either push the whole structure down or it's going to pull it up, which is going to be very, very important for our system to be stimulated. So looking over here, when we when we don't have a sound coming through, there's no sound here, nothing is going on, no movement of the tympanic membrane, nothing is coming through, nothing is coming through. We're going to see the organ of Corti, the cochlear duct is going to be in that still position. The operating point for it is going to be around a 0. So we're going to see those stereocilia standing still. They're not moving either one way or the other. However, if we have the rarefaction component hitting the eardrum, so we're starting over here, rarefaction, that means there's a drop in pressure. And when there's a drop in pressure over here, that means the pressure over here is going to be larger, and the eardrum is going to be